Hello, my name is Leah Solberg Woods and I'm from the Medical College of Wisconsin. Today I'm just going to give a brief overview of our manuscript that was recently published in Physiological Genomics. In this manuscript we used a unique genetic model, heterogeneous stock rats, to fine map a locus for glucose tolerance. Heterogeneous stock rats are unique because they're made up of 8 different inbred strains, and these are shown in this figure here. These inbred strains are then bred together for over 50 generations, such that the resulting animals are a random mosaic of these founders. So the power of this strategy is that with each generation of breeding, the distance between recombinants becomes smaller and smaller, thus enabling fine mapping down to just a few megabases. So this strategy has been used in the heterogeneous stock mouse, and they have been able to fine map hundreds of traits to an average of just a few megabases. Our paper is only the second paper to use the heterogeneous stock rat, and it's the first paper to use the rat to look at a diabetic phenotype. So the phenotype that we were interested in is glucose tolerance. In this next figure, you can see on the left-hand side glucose tolerance profiles for 5 of the 8 inbred founders. And what you can see is that 2 of the founders, WKY in red and ACI in purple, show glucose tolerance. So you can see we take a basal sample at time 0. We then inject the animals into peritoneally with a glucose, and we then collect samples up to 90 minutes after that glucose injection. So both the WKY and the ACI, their glucose goes up higher, and it takes them longer to come back to normal. You can then, on the right-hand side, see glucose tolerance profiles for heterogeneous stock rats. In this figure, each separate line, it represents a single animal. And what you can see is that the variation between the inbred strains is found within the heterogeneous stock animals. So some animals are glucose intolerant, like the WKY and the ACI, and other animals have very normal glucose tolerance profiles. And you have lots of animals that are in between there. These results simply suggest that this is a mappable trait using this strategy. Now, my group was interested in a specific region on rat chromosome 1, and this is shown in this next figure. You can see rat chromosome 1 is shown vertically to the left here from about 200 to 267 megabases. And then each black bar to the right of that represents a QTL that's been identified by a different group. Now, importantly, these QTLs have been found in different F2 rat intercrosses. So many use a diabetic strain such as the GK or the Olatef rat, but there have also been loci identified in this region using the Worcester-Kyoto rat, one of the founders of the HS, that is glucose intolerant. So it's also important to note that the homologous region in human has also been identified in many linkage studies, and recently in human genome-wide association studies, 3 of these genes fall within this homologous region in the human. These genes are not shown in this figure here. So using HS rats, we have successfully identified a 2.4 megabase QTL, and that is represented in this figure by the arrow. Next, I'm going to talk about the analysis that we use to identify this QTL, because this is another aspect that makes our paper unique. So if we go to the next figure, you can see here in the top panel, this is the scan that identifies this 2.4 megabase region. It's at about 207 megabases. So what makes the analysis unique is that we have incorporated the complex family structure of the heterogeneous stock rats into both the threshold procedure and the regression model of the analysis. So let me just back up a little bit. So heterogeneous stock rats have complex family relationships because each animal is somehow related to each other, but by different degrees. And that is inherent in the breeding scheme necessary to maintain a high degree of heterozygosity in these animals. It's been found if this family structure is not taken into account, then many false QTLs are reported. So in order to make sure we're not identifying a false QTL, we need to take these family relationships into account. Now, if you have whole genome-wide information, you can do multi-QTL modeling in order to take into account some of those confounds. However, we have only looked at a single region on rat chromosome 1. So in order to take into account these family relationships, we have developed a method that Johannesson et al. Used last year, developed it further. So Johannesson et al. Looked at family relationships, or incorporated family relationships into the significance threshold procedure. We've taken that one step further and looked at family relationships not only in the significance threshold procedure, but also in the regression model. That's what you see in the top panel. We've then compared that to what Johannesson et al. Did and incorporated family structure only into the threshold procedure. And you can see when we do that, another QTL appears at about 220 megabases. We further compared that to a significance threshold that just uses permutation and does not take into account family relationships. And you can see even a third peak that is showing up. However, we have very little confidence in that peak. We have most confidence in this 2.4 megabase peak that's located at about 207 megabases. And the future plans of our lab are then to identify a gene or genes in that region that plays a role in glucose regulation. So importantly, this 2.4 megabase region has not previously been identified using rat congenic strains, nor do any of the recent genes identified in human genome-wide association studies fall within this region. As such, we believe that by identifying a gene or genes within this small region, we will be able to identify a novel gene that plays a role in glucose regulation. So in summary, we have used heterogeneous stock rats to fine map a locus for glucose tolerance. It's a novel locus. Our study highlights the utility of heterogeneous stock rats and also the importance of incorporating family structure into the analysis.